everyone, so we are from Gotua. Would like to present about demonstration of inductively coupled plasma mass spectrometry, or known as ICP-MS, for the subject quantitative analysis and instrumentation. And our beloved lecturer is Dr. Nur Fazrin binti Muhammad Hanif. So basically, this is our group member, consists of 4 people, which is me as Nurul Amani binti Nur Azman, Muhammad Nur Hazim bin Muhammad Razali, Muhammad Faiz bin Muhammad Najib, and Harith Abdul Azim bin Ismail. For the contents, first, instrument used for ICP-MS, second, how ICP-MS works, and third is conclusion. First, instrument used for ICP-MS. On the top right of the slide, which is the Perkin Elmer ELAN 6000 ICP-MS, this is a fully computer-controlled laboratory instrument. Therefore, what are the unique features of this instrument? First, the auto-lens ion optic system for on-the-fly, computer-controlled ion optimization. The thermostatically controlled fast electronic allows for single-point peak hopping. And third, the dual-stage discrete dynamic detector allows both high and low-range signal to be measured simultaneously in a single scan. Second, how ICP-MS works. How does ICP-MS work? The inductively coupled plasma, ICP, is an ionization source that fully decomposes a sample into its constituent element and transforms those elements into ions. It is typically composed of argon gas, and energy is coupled to it using an induction coil to form the plasma. So as you can see, this is the overview diagram of ICP-MS. Basically, we will start with sample solution and end with signal conversion. For more detail, we will move to the next presenter.
Sample introduction system provide the means of getting sample into instrument, composed nebulizer and spray chamber. The liquid sample introduced to nebulizer by a peristaltic pump through self-aspiration that create aerosol fine droplets. The fine droplets are passed through a spray chamber before allowed to enter plasma. ICP torch and RF coil. ICP torch generate argon plasma 6000 Celsius. Plasma generate passing argon through concentric quartz tube, which is ICP torch that are wrapped at one end radio frequency. Energy supplied to the coil by RF generator coupled with argon produced plasma. During voyage into plasma, liquid droplet dried to solid, then heated to gas, then absorb more energy, release one electron, form singly charged ion. Singly charged ion exit the plasma and enter interface. The interface sampling ion allows plasma and ion lens system to coexist. Allow ion generated by the plasma to pass into ion lens region. Consists 2 or 3 inverted funnel-like device called cone that need opening about 1 mm. Downstream focusing ion beam is required by use of single series of charged device called ion lens. If 3 cones are used, then there is no need for ion lens.
Thank you to the previous presenter. Now we will move to the vacuum system. Vacuum system is used to provide correct operating pressure. This is because ions need to not collide with gas molecules during the travel. So gas molecules are removed by using the combination of the pumps. They use 2 roughing pumps and 2 turbo pumps, which is large and small. The large turbo pump is used to maintain low pressure just behind the interface, and the small turbo pump is used to maintain super low pressure at the mass analyzer and detector. The roughing pump used to remove gas from between the sampling and schema cones. Next is ion deflection device. This is before ion enter the mass analyzer. They use quadruple ion deflector or QID. Ion beam exiting the interface contains some non-ionized material, which is neutral and photons. So QID is used to separate ions from neutral and photon. The ions are turned by the quadruple by right angle for their entry into the filtering quadruple or universal set. If you look into the picture, the straight line is photons and neutral, which is indicated by green color, and the blue color is ion. The next is the mass spectrometer, or you can call it by the mass analyzer. The mass spectrometer separates the singly charged ion from each other by mass, acting as mass filter. The quadruple works by setting voltage and radio frequencies to allow ions of selected mass to charge ratio to remain stable within the rods. So, the selected ion will pass towards the detector while the other ions are ejected and collide to the rods.
Assalamualaikum and very good morning. My name is Hari Razim Ismail, and now I would like to talk about data handling and system controller of ICP-MS. The software in ICP-MS translates the ion count measured by the detector into information. Proper ignition of the plasma, pressure within the high vacuum region, and the voltage applied to the detector are constantly monitored by the controller. ICP-MS provides data in four ways: either semi-quantitative analysis, quantitative analysis, isotope dilution analysis, or isotope ratio analysis. Here is an example of how ICP-MS presents data.
The last is conclusion.
In conclusion, ICP-MS is highly sensitive analytical technique for determination of trace element of clinical interest in biological fluids. ICP-MS offers numerous features which can make it particularly attractive for the clinical laboratory. These include high sensitive, wide linear dynamic range, wide elemental coverage, and multi-element capability. Pros and cons of ICP-MS. The advantage of ICP-MS is fast multi-element analysis, wide range of element analysis, and isotopic analysis capability. The disadvantage is total dissolved solids in ICP-MS is less than 0.2%, high running costs, and more operator experience needed.